Assalamu alaikum and welcome again friends to our channel BIOCHEM graduates. Here is another video tutorial or lecture about the introduction of proteomics. So first of all, let's start our lecture, but don't forget to subscribe our channel and click on the bell icon. Thank you. So in this lecture we are going to discuss about the introduction of proteomics and why proteomics is important, or what is the aim of the proteomics, or the techniques through which we identify the structure or the protein interaction, like 2D 1D or 2D electrophoresis, mass spectroscopy, and many other techniques. So we'll try to understand and we'll try to cover all these topics in our this lecture tutorial. So let's start our lecture by the introduction of the proteomics. So we know that the proteomics is a huge topic, or a huge part of the modern science or modern scientific society, which combine biological tools and biological scientific tools like we discuss in RVC in bioinformatics, to get understanding of about the proteins and their interaction between the other proteins or between other chemical bondings and all that. So proteomics is a large scale study of proteins. We know that the proteins are a vital part of living organisms with many functions. So the word proteome means the proteome is the entire set of the protein that is produced or modified by the organisms or system. Like we say in genomics, what is genome? Genome is the entire set of the genes. Like that, proteome is the entire set of the protein. So when we see in the proteomics has enabled the identification of increasing number of the proteins. This varies with time and distinct requirement or stresses. First of all, when we want to understand the concept of proteomics, we should know about the central dogma of biology. So we now know that the central dogma of biology, like we have DNA, and DNA is transcripted into RNA, and RNA further translated into the proteins. So this is the central dogma of life. So we know that the study of DNA, we are talking about the DNA, it's called the genomics. And when we discussed about the RNA, we are talking about the transcriptomics, okay. And because we are talking about the transcription. And finally, when we talk about the protein interaction, their function and their 3D structures, that means we are talking about the proteomics. So now we know that proteomics is important, but question is why proteomics is important or useful, or what is the aim of the proteomics. So we all know that the mammalian system are much more complex than it can be decoded by their genes alone, and the biological command of the organisms are largely controlled through the function of the proteins. We know that the human genome contains the complete set of genes required to build a functional human being. However, the genome is the only source of information. In order to function, it must be expressed. We know that the transcription of gene is a first stage of gene expression, and it is followed by the translation of a messenger RNA to produce protein. Here we are again talking about the central dogma of our life. DNA contains a set of genomes, and then it is transcribed into mRNA, and then it is translated into the proteins. The proteome is the complete set of protein produced by the genome at one time. The proteome is much more complex than either the genome or transcriptome. This is because each protein can be chemically modified in different ways after synthesis. Like many after the modification or synthesis, many proteins have carbohydrated groups added to them. Others are phosphorylated or acylated or methylated. So it all depends upon the modification or post-transcriptional modification of the protein. The proteome is also very dynamic. Most of our cells contain the same genome regardless of our cells' development stage or environmental condition. The proteome varies considerably in these differing circumstances due to different patterns of gene expression and different patterns of protein modification. Why proteomics is important? Okay, proteomics, the study of proteome, we know that is important because of protein represent the actual function in the functional molecule in the cell. When the mutation occur in DNA, it is the protein that ultimately affected. Okay, drugs, when they have for example, when we targeted the protein with different drugs, so these when they are beneficial effects, they have beneficial effects do so by the interacting with proteins. So proteomics therefore covers a number of different aspects of protein function, including like a structural proteomics. So structural proteomics is the large scale of analysis of protein structure. Protein structure comparisons can help to identify the function of newly discovered genes. Like structural analysis can also show when drugs bind to the protein, where the protein interact with each other, are the point where the interaction point of the protein. When we target the drug there, so we can understand the structure of the protein, and we will discover the behavior or function of the we identify the gene. And these techniques, this is achieved by using the technologies such as X-ray crystallography, NMR, or other spectroscopy methods. Okay, when we going through the comparison method, or when we want to compare the some disease with healthy one, or some affected tissues with the healthy one tissues. So in this method, this means we are going to do the expression proteomics. So the expression proteomics, the large analysis of protein expression, this can be helped to identify the main proteins found in particular sample and proteins differently expressed in related samples, such as diseases versus, as I have told earlier, that like diseases versus healthy tissues. A protein found in diseased sample may be represented as a useful drug target or diagnostic marker. So we can express, or we can identify these expressions through the Mantia to the PEG or electrophoresis or mass spectrometry are used here. The third number, which is very important aspect of the proteomics, is interaction of protein-protein interaction. When we study through the proteomics, our protein, nature of protein and their functions, and so it must be important to identify or to know about the interaction of the protein or the interaction of protein-protein interactions. So interaction proteomics, the large scale of analysis of protein interaction. Okay, the characterization of protein interaction helps to determine protein function and can show how proteins assemble in large complexes, or how proteins assembles means how they are formed from primary structure to secondary structure, from secondary structure to tertiary structure, how they form their complex histories, how form their 3D structures. Okay, we can identify their protein interaction by different technologies like affinity purification, mass spectrometry, and G2 hybrid system. which are particularly useful for their interaction purpose, for to know about their interactions of proteomics or interaction of the proteins. Here we must know that the techniques for proteome analysis are not as straightforward as those used in the transcriptomics. But the advantage of proteomics is that the real function molecule of cell are being studied. It must be mentioned, or it is necessary to say that the strong gene expression resulting in the abundant mRNA does not necessarily mean that the corresponding protein is also abundant and indeed active in the cell. Okay, it is not necessary that some mRNA are transcripted, so it must be corresponding protein is also abundant, or it is indeed in the indeed it active in the cell. It must be maybe it may be inactivated and stored in somewhere, or maybe it simply dissociates and perform not any function, so it is not necessary. That transcripted mRNA have to form the every one of us know the one concept of one gene and one protein concept, but it is not valid nowadays, or it has become invalid. Why? Because one genome can have many proteomes. That means one gene is no longer equal to one protein, but one gene have many proteins. Here we must remind that we are talking about the eukaryotic system or eukaryotic cell, not proteomic cell. Though this statement, one gene, many proteome, is valid for the eukaryotic cells or eukaryotic organisms. So, from one gene in eukaryotic system, we can produce many proteins, not one protein. Why we say that? Because we are saying that because when DNA is transcripted, it has many stretches of polypeptide chains. We have one gene and it is encoding particular polypeptide chain sequence, but gene can be manipulated in many different ways, and many different ways gene modification or amplification can be established. For example, simple mutation can be occur. Translation, different translational modification can be occur, and obviously post-transcriptional modification can part of this. RNA editing or RNA splicing can be part of it, and many more amplifications or modifications can occur. So when we go through from the central dogma of life, which is DNA, and then RNA and the protein, for example, DNA, which carry only the gene sequence or genomes, then and RNA comes from the transcriptional method, and after transcription there will be translation, and post-transcriptional method the protein will be produced. So that is the central dogma of life, and many modifications occurs and many changes, amplifications occur at every stage of the life. At every stage of this dogma, or every stage of these modifications and protein sequence, protein translation. Because DNA containing only one information, but manipulation of these sequences and these information can be amplified into RNA or further more advanced, from which RNA will be produced. RNA produced in the eukaryotes is not totally that RNA which is translated or produced into the are enabled to the produce protein, because RNA can also have many different compounds or containing many other things. Then the sharpening or the splicing, editing of the RNA will occur in the post-transcriptional method, and then protein will be produced. Even once the protein is produced, there will also be the post-translational modification occur in that protein. So they have concluded the conclusion is that when we are going through the DNA, then the RNA and the protein, the chances of the diversity, the chances of the variance increasing at every stage. So this protein will produce, and this protein will show the different functions, different expressions, and different part of the cell, human body, or different functions in organisms, like and shows different expressions like nerve cells shows different expression in the form of nerve impulses, and liver muscle cells shows the different functions or different expressions in muscle cells or in our liver cells. So we can say that where at the DNA level we have only 4 bases, okay. DNA sequence has 4 bases, but as we going through the DNA to the protein, and at the protein level we have 20 amino acids. So it is clearly shown that diversification are the variance from the DNA to the protein. So whereas the genome of the all organisms is essentially invariant in all its cell, proteomes vary among different cell types and also vary with time. This is because with time any protein product of a single gene may exist in multiple forms because post-translational modification, the existence of mutants and the formation of splice variant. Now, why proteomics is important, or why we are studying proteomics? Because we can simply find one sentence that genomics do not have answers to all of our questions. Our only single gene don't have the all information which is obtained through the protein structure, protein modifications, or protein functions. So let's take an example. For example, we have DNA and it is transcribed into RNA, and it will be processed into the mRNA through the post-transcriptional modification, alternative splicing, editing, okay, and polyadenylation. But after this mRNA, this mRNA is translated into the protein through the translational regulation, and this protein will be further modified into the proteolysis, post-transcriptional modification, compartmentalizations. But we can see that every stage here is that transcriptional or translational modifications are occur at every stage or every step. For example, chance of modification is transcriptional regulation, then alternative splicing, mRNA editing, and polyadenylation. At the RNA, from processing RNA to mRNA, then from mRNA to protein, there will be translation and translation regulation, and then compartmentalization after the protein synthesis. So these are the chances of the modifications or chances of the variant occurs at every stage. So we can say that behavior of gene product is difficult or impossible to predict from gene sequence or predict for looking at just DNA. Because when we look at the protein, this protein is thing which will going to predict what will the cell is going to perform, or what the cell is need. Gene is similar to all these cells, but the only varying thing is from cell to cell is that protein. So the protein in some cells are expressed more, and their particular function is expressed in some cells more efficiently or more dramatically than the other cells, in which the abundance or the quantity of that protein is small quantity, and show their functions in less amount, are giving a different function from that protein, which is showing the different functions in different cells. So we can say that even if gene is transcribed, its expression may be regulated at the level of the translation, at the level of the protein synthesis. Protein products are subjected to further control by the post-translation modification. Okay, as I have mentioned this earlier, that So here we are able to say that the proteomics is the field that promises to bridge the gap between genome sequence and cellular behavior. What does that mean? We know that the genome was described or the studied before the proteomics, and proteomics is new field and a diverse field. So there was a difference between the gene and the cellular behavior. So proteomics is the field, or it performs the bridge between this gene sequence and the cellular behavior cannot be or never can be explained by the just looking at the genomics. So for that we have to fulfill this gap, and by the proteomics study. Proteomics is now helping us to give these questions, unanswered questions. We are better understand or better explained by the proteomics. So we can say that the proteomics can answer about the protein identification, protein expression studies, protein function, protein post-translational modifications, and of course the most important thing is protein-protein interaction. We can classify the proteomics into three general steps or three general categories, depend on its level of complexity. So these three classifications, three stages or three steps are classified on the basis of their complexities. Okay. Now, number one is protein expression comparison. Okay, which can be easily detected by some protein comparison analysis like microarray analysis. So it's just the beginning of proteomics. The second one is structural proteomics, which is complex or more diverse than the first expressional comparison, and that is structural proteomics. The goal of this structural proteomics is to map out the 3D structure of proteins and protein complexes in a large using bioinformatics tools. This proteomic part is highly combined with computer science and bioinformatics tools. Otherwise, we cannot establish our goal. And after that, we have functional proteomics. In this, we have the study protein-protein interaction, 3D structure, cellular localization, everything, or in order to understand the physiological function of the whole set of proteome. This is most difficult one, so this means the study of everything. So we can say functional proteomics is most difficult one or most diverse one part of the proteomics. So now which techniques are utilized in the study of the proteomics? We have the different techniques which are utilized to understand the interaction of these proteins. So for the functional proteins, and the first step is protein isolation. For example, we have a scale from which we extract the proteins by different cell extraction process techniques or different steps of cell extractions. Maybe through the electrophoresis or other method. Then we have to quantify this protein and then digest this protein. First step is we have identify the protein, then we have to digest that protein through the 2D electrophoresis. We can quantify the protein through the 2D or 2-page electrophoresis, and then we have to digest the protein through the tryptic process, and we further proceed these proteins to the mass spectrometry. Then we after the mass spectrometry will identify these proteins through by using bioinformatics tools, and then the quantification will be occur. Okay, so here the protein isolation is completed. Second step is separation or through the 2D gel electrophoresis. So once we have isolated the proteins or separate the proteins, we have the another separation step, which is rely on this this 2D gel electrophoresis. So in this 2D gel electrophoresis, what we do, we simply load the sample of our protein to separate those proteins, and we get our desired different types of protein. We can get the different types of protein depending on depend on their molecular weight as well as on their isoelectric point. Once we got our protein separated out through the 2D electrophoresis, then the third step is identification through MS analysis. MS, mass spectrometry analysis. Okay, third step is identification. So third step is analysis or identification to know about which type of protein or what protein we are dealing with. So for that, so in this we have two samples and we load into our mass spectrometry. We provide them mass spectroanalyzer, and we get our data. So by looking at data, we can identify it, or we can come to know which protein we are dealing with. If it is known, we can identify it through this technique, and if it is even unknown, we can also identify those through this technique. Once we analyzed this protein through the mass spectrometry, we further proceed to the interaction studies. So the fourth step is the interaction studies. Till the mass spectrometry, we just identified our protein, but now we had to see the interaction of these proteins. So how can we find these interaction? So there are several techniques to find out the protein-protein interaction. So these protein-protein interaction can be found by two different methods. Number one, which we discussed just a moment, is antibody array. Here I should mention that this interaction can be studied in vitro as well as vivo studies. In vitro study, we can see these interaction in yeast to hybrid. In vivo studies, we can see this in two hybrid. In vivo studies, we have yeast to hybrid system, but in vitro study, it can be achieved by the antibody arrays. Okay, obviously the immuno system it says. Okay, so through this technique we can obtain good low abundance proteins, but there will be the issue of the antibody specificity, because we know that when we are going through the antibody assays, we have the specificity issue. The second type of interaction studies is array-based protein interaction detection. Array-based protein interaction detection. It is also in vitro analysis. In this we have desired probe, and in this technique the protein is bind with the desired probe. So as it will bind with a different desired probe, it will give different signals in the form of different colors. So, and in vivo studies of protein interaction, which is the third type of the protein interaction, is two hybrid analysis, or also known as yeast to hybrid analysis system. And fifth, the most important part of the protein interaction, protein identification, is the constructing the interaction maps grid. Okay, for example, after the interaction we have got different types of proteins, and now we have to draw a map of these proteins according to their interaction. To construct this model, different proteins are joined according to their function and the protein close to one another. So here we can obtain a graph or a map of the protein. So this is all about the proteomics. Now in the next lecture will be the 1D and 2D gel electrophoresis. Hope you like the video. Don't forget to subscribe our channel, share the video, and click on the icon button.